So I'm very happy to introduce Shaun Brothers. Shaun and I have been working together pretty intensely the last couple of years trying to create a more integrated drug discovery platform for the medical school. He's an associate professor in psychiatry and an associate director of the Center for Therapeutic Innovation. He's also a member of the Cancer Center and the Department of Pharmacology. He earned his Ph.D. at Oregon Health and Science University, and he also has an MBA suggesting that he's interested in entrepreneurial kinds of activities. He was a postdoc at Scripps, Florida, and then he stayed on as a scientist there for a while, and then he moved to the University of Miami in 2011 when he joined the Department of Psychiatry and was one of the co-founders of the Center for Therapeutic Innovation, of which he's the associate director, I guess. He's been involved in some startups. He continues to be involved in startups and trying to commercialize various drug discoveries. And so that's something that lots of people in the Miami project are interested in. And so I think his expertise could be helpful for us as we try and move our things through the various pipelines. He's been very productive scientifically with over 50 peer-reviewed publications. He's been a PI or a co-PI on grants from NIH Mental Health Institute, NINDS, and NIDA. He's had a number of foundation grants. He's the founder and director of the Cancer Center's Molecular Therapeutics Shared Resource, something that you might hear me babbling on about the MTSR. That's what that means. And I think it's impressive that has a successful IND submission, I guess, for the project that he's going to be talking about today. So he has diverse interests in mental health, pain, and cancer, and this Mucopolysaccharidosis disease. And so this is a kind of a disorder that's part of a larger group called lysosomal storage diseases. It's actually something that I've heard a lot over the years from my wife also. And these large carbohydrates are also known as glycosaminoglycans. And so something people here in the Miami project are interested in, especially in injury sites in central nervous system. In this particular disease, they aren't metabolized correctly and they build up in cells, including in the brain and spinal cord, and they can disturb mental development, among other things. So I'm looking forward to hearing Shaun tell us about this long-running project that he's been involved in. Take it away, Shaun.
Thank you so much, Vance. And yeah, I do a lot of different diseases, and I think the unifying theme for them all centers around therapeutic development or identifications of probes to further scientific pursuit. So yeah, I'm in cancer and other things, but my real passion is in neuroscience, and so that's why I'm in psychiatry, and that's where I like to do my work rather than have to do my work. Yeah, so Vance, good job on pronunciation. So mucopolysaccharidosis is the disease indication we're going to talk about today, and we're going to go through a project where we identified a small molecule that we're developing as a therapeutic for this lysosomal storage disorder. I do have a conflict of interest disclosure. Jupiter Neurosciences owns the intellectual property for this and is commercializing it. And I'm a founder and shareholder. So, and as Vance said, mucopolysaccharidosis type 1 is a disorder where there is a deficiency in the iduronidase enzyme that cleaves the glycosaminoglycan sugar residues. And so when you don't have this enzyme, these gags build up in the cell and just cause all kinds of chaos, mostly inflammation from cell damage, but splenomegaly, cardiomegaly, all kinds of nasty stuff. And in fact, there are a number of different mucopolysaccharidoses diseases where there are different enzymes in this metabolic pathway that are disrupted to cause accumulation of gags. And they all have a very similar phenotype where, at an early age, depending on severity, the child will appear largely normal, but by the age of 3, it's pretty clear that they're having developmental delays and things like that. It is an autosomal recessive disease. It's known as several different things. MPS type 1 is what we call it, but Hurler syndrome, Hurler-Schie syndrome are also all known. I'll go into those a little bit more because they actually have some implication. It is a rare disease, 1 in 100,000 births, although with newborn screening, that's picking up a little bit and becoming a little bit more prevalent. As I said, there are different types of the disease. So we actually think of this as a spectrum disease where we have a more severe and more mild phenotype. The more severe cases, we term them Hurler syndrome, and on the mild side, Schie syndrome. So if I use those terminologies, that's what that means. And essentially, with Hurler children, they get the disease manifests at a much earlier age. Severe developmental delay, severe mental retardation, severe issues with respiratory cardiovascular system. And essentially these kids will, if left untreated, die before the age of 10. On the Schie side, these children actually will go on to live almost a normal life. They can have a normal lifespan. The physical issues are less progressive, the information is less progressive, yet they still do have problems. And that's an important component of what we're going to be talking about today, because there is an unmet medical need within the community. Even these kids will have issues that could cause death, and generally they don't actually live a full lifetime. And then in the middle are the sort of the gray, the Hurler-Schie syndrome kids, where they have some or other of the two sides. There are a number of treatments currently for mucopolysaccharidosis. The most prevalent treatment is enzyme replacement therapy. So this is a $400,000, $500,000 actually a year treatment. The families, when they get a diagnosis of MPS type 1, they call it a bankruptcy diagnosis because the treatment is so expensive. Essentially, nobody can afford it, and they declare bankruptcy. So it's less than ideal. There's a bone marrow transplant and human stem cell transplants that also do very well. These are high mortality procedures, so not all of the MPS 1 patients go through these. The Schie syndrome kids tend to avoid this, but those are out there. They are helpful. Enzyme replacement therapy and the stem cell transplants and others are helpful for the patients. They will replace some of the enzyme in the body, but the issue is what you're putting into the body is a large protein, and it can't get to all of the places where you need it to get. So you have issues with certainly the CNS brain issues. You have joint pain, you have impaired vision. And in fact, this fellow here is named Ryan Dant, and the Ryan Foundation has supported some of this work for Ryan Dant is the first ever patient to get enzyme replacement therapy. I think it's something like 20 years ago. So he's been on ERT for 20 years. He has a diagnosis of Schie, and he's obviously quite a bit older than 10 years old. This is his wife, his mother. But Ryan still has problems. If you look at his hands, he has arthritis in his hands. He has corneal clouding. He's not able to drive because he's been essentially listed as blind. So Ryan, even though he's been on ERT for such a long time, still has unmet medical needs. And this is an important component for what we're trying to do, because we'd like to address those needs in whatever way we possibly can. So this is the premise of the approach we're taking, and essentially it boils down to looking at the two ends of the spectrum. And so if you look at this Hurler, I said, disease onset is very early, lots of cognitive impairment, death, whereas Schie is much more attenuated. In a few years back, these folks did a study where they looked at residual enzymatic activity levels in Hurler patients and compared them to Schie patients. And if you look at this, 0.2% remaining enzymatic activity versus 0.8%. And to me, that is an ultra slim margin. Like you could look at that and say, well, there's no difference. And you could essentially be right, except that these kids will die when they're 10 and these kids will live a long lifetime. And so really, we didn't feel like the bar was that high to want to be able to go from such a small number to a much larger number. The other thing to recognize here is that even in the most severe patients, there is some remaining enzymatic activity. And through other work that I'm not going to show you, it appears that having absolutely zero activity is probably more along the lines of embryonic lethal. You need a little bit to be able to survive. So what our idea is can we get this number to be higher? Can we get it to be as much as 0.8%? Or can we get both of these numbers to like something like 1%? We think 1% is kind of the magic number where you wouldn't even see any symptomatology. So we were approached by a family who had a child with MPS 1. She is actually doing well. She's in the side of the spectrum. And that family sponsored the early work for compound identification. Essentially, we took her cells, her fibroblast cells, and screened a number of different libraries using a real-time PCR assay. It's the cells CT assay technology, which we adapted to a 3D 4-well format and a no-wash format. And we actually worked quite hard on this, and Claude Volmar was the one who really enabled this. We did such a good job that actually the company came back to us and said, hey, how did you do it? Because they weren't able to get to that point. Essentially, what we were looking for is an upregulation of RNA. So the idea is that we have a little bit of remaining enzymatic activity in the cells, and if we can just boost that a little bit. So we were looking for things that would boost enzyme protein production by way of boosting RNA production. And so that was the idea behind the screening. The immediate obvious candidate for boosting the RNA was this molecule resveratrol, which I'll talk quite a bit about. We did an independent RNA-seq confirmation of this assay to show that yes, very high levels of resveratrol would indeed boost RNA production. So resveratrol quickly became the focus of our studies. We then tested resveratrol in these same cells for enzymatic activity. So the previous one was RNA, here we're doing enzymatic activity. The assay is quite simple actually. So we have this sugar moiety attached to this fluorescent moiety, and this alpha iduronidase enzyme that's deficient in the patients would cleave this and shift the fluorescent spectrum, and we can pick that out. So it's what we call a 4-MULE assay. Essentially, we had resveratrol, we had another couple of still being analogs. This is resveratrol. Pisatinol was also a heavy hit of ours, but we had other molecules in the same structural class that we tested. And you can see that, without treatment, there's no measurable enzymatic activity. These are normal wild type cells, so it's, kind of massive. Resveratrol, pisatinol, and all of them increased enzymatic activity. And so we were quite excited to see, that increase. This one, you did a really good job. Unfortunately, this one is used to make vinyl. And so it was a chemical that just wasn't tractable for therapeutic use. And so we quickly focused on resveratrol and pisatinol. With resveratrol being available even over the counter, it was an obvious one to take into further development. We tested the effect of resveratrol on 4 different MPS cell lines. So these are from four different patients. This is in addition to the original screening cell line. And you can see their mutational statuses for their enzymes where, they have sort of different mutations. In all cases, we had some effect of resveratrol, although that effect varied. And actually, it kind of made sense, this data, because if you look at, for example, this 6-year-old boy, his termination codon was actually changed into a cytosine, and so, a cysteine. And so he has the full-length protein plus a few amino acids on the end, which killed the enzyme. We think it's probably degradation related. And with resveratrol, we were able to really rescue that quite nicely. This girl has some very severe deleterious mutations where an early termination codon or a mutation within the enzymatic cleavage site really caused a reduction in her hyaluronidase activity. And so we still got a little tickle of effect with resveratrol in this girl as well. But suffice to say, we were happy to see, beyond our initial screening effort, that we got those kinds of responses. And it
John,
does
we have a question that seems interesting. So does I can't pronounce that correctly, so I'm going to let you do it. But does this is there some specificity in the increase in the mRNA for this enzyme, or is this a global increase in transcription, or what's going on there?
Yeah, it's both specific and non-specific. So resveratrol is known to activate sirtuin enzymes, which are epigenetic modifiers. And so we think there's a global epigenetic reorganization that occurs with resveratrol, which happens to increase the hyaluronidase RNA. We've actually, as part of that RNA-seq and other efforts, have looked at other enzymes in that pathway. And so, for example, for the MPS II enzyme, it does not increase that RNA. So there is some specificity to this gene, but it is a global epigenetic modulator. So there isn't specificity in that way to that specific gene product.
Thanks.
So of course, the obvious question is there an effect on glycosaminoglycans? Now, just as a forewarning, glycosaminoglycan content in cells, even with enzyme replacement therapy or in the body with enzyme replacement therapy, they really don't vary that much. So we weren't expecting to see a huge change, but nonetheless, in the patient's cells treated with resveratrol, we did see a decrease in glycosaminoglycan content. Here now we've taken wild type mice and treated them with 50 mg per kg just to get a sense for the in vivo potential in vivo effects. And we did see an increase in RNA expression in these mice. So we have some evidence that it will not only correct the major molecular underlying defect, but also be useful in an in vivo situation. There is an MPS I mouse model. It's a W392X double knock-in, which is synonymous with the W402X in humans. We injected the resveratrol at 50 mg per kg for 2 weeks into these mice and saw that parameters associated with the MPS I phenotype were altered. So for example, grip strength here is shown to be higher with resveratrol test, with resveratrol injection. And this grip strength test is essentially where you take a metal grid, you let the mouse sort of attach to it, you turn it upside down over a dark background and let the mouse sort of fall. And because it's dark, it's in the dark and the mouse can't see the floor, they sort of hold on for dear life. And so the vehicle treated mice held on for less amount of time, indicating less ability to grip or maybe some issues with their musculoskeletal system, whereas the resveratrol mice held on for longer. So, again, just showing that there is some potential for therapeutic benefit in a patient population. Okay, so as sort of went over this earlier, this is introducing resveratrol. It's a low molecular weight compound. It does activate the sirtuin 1 system, but it also activates a number of different systems. It's really quite a dirty drug, but we feel like as long as it does what we want it to do, and as long as it's safe, it was something that we could continue to develop. There are over 14,000 publications on resveratrol. So pretty much if you think that it, if you have an idea for something to test, it's probably already been done. And actually, this was a great boon for us as we were developing this, because we could take hints from the publication and the literature and really fine tune our development process. It's been tested in clinical trials, and I would say the vast majority of these publications, The major underlying issue with resveratrol is that it's underdosed. So resveratrol has a significant first pass metabolic issue. It gets chewed up by the liver, and very low levels will actually hit the bloodstream. So you can see that here. If we give an IV dose of resveratrol, we get quite a lot of it going into the bloodstream, obviously. But if it has to go through the liver in an oral administration route, we just can't get nearly as much. So it's something like 0.5% resveratrol hits the bloodstream if you give it orally. So that was our problem. We said, okay, if we want to treat MPS I patients with this, we're gonna have to figure this out. So the bioavailability issue, it wasn't unique. It wasn't something that we were the first to encounter. Other people have been dealing with this over the years. The other issue with resveratrol is if you give a high dose, and we're talking like 5 grams a day, you get GI effects, and that's probably a bulk action, issue, water entering the gut and diarrhea. So limiting the GI potential for side effect was also a problem we were trying to address as we were going through this formulation. We looked at what had been done so far, and the best available formulation for resveratrol was this micronized form that was developed by Friedrichs back in 2006, 2007. And essentially they grind the resveratrol up to a 2-micron size. And I'm guessing here, but my guess is that it dissolves very quickly in the gut, enters the liver sort of all at one big whack, saturates liver enzymes, and that's why you're getting a little bit more bioavailability from the micronized than you would from this, like a solid pill, where it would take time to dissolve and sort of trickle into the liver. We weren't happy with the bioavailability from this micronized version. Even here, we were looking at between 2 and 5 gram doses per day. And, in children especially, that becomes quite, untenable fairly quickly. So we actually invented a new formulation for resveratrol. It's essentially a micellar formulation. So everyone's familiar with the liposome, where you'll have an interior that's sort of hydrophilic, and in a micelle, what you have is an interior that's very lipophilic, and resveratrol itself is very lipophilic, so it sits nicely into this micelle. So this is sort of an animation of what that would look like. And actually, as we're developing this, we learn here and there. We actually think that there's a good portion of this that enters the bloodstream through the lymphatic system, and so it completely bypasses the liver. So that's what the micelle looks like. Here's a solubility test. And so the left-hand side will be sort of unformulated compound. This is actually turmeric, but it's the same thing for turmeric as it is for resveratrol, where on the right side, we will have formulated this, and you can see it just goes straight into solution. So finally, now with this in hand, and this is now, I think, something like 8 years of work on this project, we felt comfortable in taking this forward and doing some more clinically oriented development. We were calling the formulation Jutraw. It was Jupiter Orphan Therapeutics, now it's Jupiter Neurosciences. And this is with the company. This is data that we've generated with the company. Here you can see we've taken this micronized version of resveratrol and compared it to Jutraw, and looking at how much gets into the bloodstream after an oral administration. And this is what we saw before, very, very low amounts of resveratrol getting in, whereas with this Jutraw formulation, with the exact same dose, we get a much higher tenfold increase in the amount of resveratrol that hits the system. We do get brain penetration. This is often asked. It does get into the brain, although not at 100%, 23%. We feel like any brain penetration of the molecule could be beneficial to the MPS I patient population, because especially the CNS is a major issue for enzyme replacement therapy, because ERT doesn't get into the brain at all. We validated the effect of the Jutraw formulation on iduronidase, and this is in wild-type rats, and essentially looking at iduronidase activity. So our micronized resveratrol, did increase iduronidase activity. This is the normal wild-type level of iduronidase activity, so we're actually getting quite a lot. And then here we're looking at the Jutraw effect, and it's obviously quite a lot more than the micronized resveratrol, likely due to the increased exposure. Yeah, this is it's a pretty interesting slide, I'd say. The ethics of taking your own drugs is somewhat gray, but it appears that it's not entirely frowned upon. Everything that's in the Jutraw formulation is available over the counter, so we took it and we measured our concentration of resveratrol that was in our bloodstream, me and two of my colleagues at University of Miami, and looked to see whether that Jutraw was actually getting into our bloodstream at a significant amount. And you can see with three of us taking it, it got in quite a lot. Again, further suggesting that there is some potential for clinical utility. Okay, so now we're doing a much more controlled study. As I said, with the 14,000 publications, quite a number of clinical trials have been run. There were really two clinical trials that we felt were of high enough quality and appropriately dosed to really kind of pay attention to. This one is what we call the Turner Alzheimer's clinical trial. So resveratrol has been suggested to be useful for Alzheimer's disease. Scott Turner at Georgetown essentially took 120 patients and dosed them over the course of a year in a dose escalation trial, where the final dose was 2 grams of resveratrol. This is unformulated resveratrol for 13 weeks. I contacted Scott and got a hold of some of his plasma. So he gave me 20 plasma samples from this trial. And we measured iduronidase activity. So this is a well-controlled trial. And notably, the plasma concentration was around 200 nanograms per mil. So, if we're looking at 1,000 nanograms per mil versus 200 nanograms per mil, the Jutraw, again, is looking quite promising. The iduronidase activity is strikingly high. I mean, it was just massive. This is a logarithmic axis here. And so it is an enzymatic activity assay. So maybe logarithms aren't so bad, but it was just huge. We didn't expect so much. I will note that Scott Turner observed GI issues even with 2 grams a day for resveratrol. And then certainly after 52 weeks of treatment, with this big effect, it looks like the chronic treatment may be of more benefit than just that single dosing that we've seen in some of the animal models. The other clinical trial that we, and I should say, Scott remains close to this project, and we're working with him on submitting grants and things. So he's on board with using this formulation in his Alzheimer's cohorts. The other high quality data set came from Murdoch Children's Research Institute in Australia in a Friedrichs ataxia trial. Essentially, Martin Delatecki, who ran the study, is also working with us and wants to get a hold of Jutraw for his Friedrichs ataxia patients. Essentially, this was an open label study of unformulated resveratrol, and the Friedrichs ataxia patients were given 5 grams daily of resveratrol. So just whopping huge amounts of dosing. Obviously, GI effects were observed, and he saw positive effects. And so if you look at this, again, the caveat here is that it's an open label trial. But if you look at this 5 gram daily dose in a Friedrichs ataxia rating scale score, there was a nice p-value, modified FARS score, and some other areas like, for example, hearing and speech, all had positive responses to this. And we've been advised that if we can get even one of these to repeat with Jutraw, we'll have the basis for an approval for the drug. So obviously, we're looking to do Friedrichs ataxia as well. And I'll note that the circulating plasma levels here were around 300 nanograms per mil. What's interesting about this trial, and maybe a little bit more special, is that I lied to you. Martin Delatecki gave 5 grams daily, but he also had another cohort that received 1 gram daily. So he had that second comparison. I don't have the data here, but in 1 gram daily patients, none of these p-values showed up. This was really important for us because it essentially gave us a minimum baseline for circulating plasma level that we wanted to hit with our Jutraw formulation, where we expected to see an effect. So 300 nanograms per mil now becomes our target because we know in a clinical setting, Martin was able to see that a clinical benefit from that level of circulating resveratrol. So, again, just to outline where we've come with this premise, so we have this internal study at where we measured very high levels of resveratrol with Jutraw with 500 milligrams of resveratrol. This is the Turner study with 2 grams of resveratrol, not very high, and then this is the MCRI study, where they got a little bit more. So if this clinical outcome replicates in like a phase 1 trial, then we really have a product that can beat out unformulated resveratrol. So the idea, again, can we get these numbers to jump? We think we can based on cellular data. We think we can get it into the body at sufficient concentrations to be able to achieve these kinds of effects. So this was really quite nice data to support clinical trial, essentially. So we went to the FDA. Here you can see this is a test manufacturing of Jutraw. We had the pre-IND meetings completed. This was in person. Actually, the FDA took quite a lot of interest in this program. We think because the low toxicity potential for resveratrol, they're really excited about that potential for turning it into a drug. They don't have to worry about toxicity so much. We were allowed to open the IND with a phase 1 PK study, and we didn't need to do additional toxicology. But the FDA did give us guidance on exposure levels. We weren't allowed to surpass this exposure level, 2.1 micrograms per hour per mil. We did discuss with the FDA on outcomes. So the previous clinical trials in mucopolysaccharidosis patients were focused on 6-minute walk distance, and these were the basis for the approval for enzyme replacement therapy. FDA wanted us to also do 6-minute walk, but the issue is, and we work with patient foundations, they've advised us that no patient will come off ERT in order to join a study with our drug. So we were sort of in a position where we felt that the 6-minute walk test would not be reflective of any improvement that the Jutraw could impart. So, back and forth with the FDA, we essentially got them to agree to different endpoints. So things like range of motion, pain, and a quality of life score are actually those that are on our radar. It'll be an orphan drug designation and a priority review voucher, but we ended up getting the IND, which was pretty nice. This is the design of the phase 1 clinical trial. It was actually, we got an SBIR, and it was paid for entirely by the National Institute on Aging for the Alzheimer's indication. The trial was with 24 healthy volunteers, dose escalation trial with originally planned to be 200, 500, and 1,000 milligrams, although we hit our exposure maximum, so we actually had to drop this higher dose down to 700, which was fine. We were looking to measure whether a high-fat food diet, because of how lipophilic resveratrol is, would have an effect on exposure, and to measure the sulfuronidase and glucuronidase metabolites of resveratrol as part of the trial. So this was done during COVID. It was very interesting to try to coordinate this. The patients had to come in for 24 hours before the 24-hour trial in order to do the COVID-related stuff. So they were kind of cranky by the time they got to the trial. And in fact, our adverse events were things like headache and sleepiness, which would be more likely to do with the COVID procedures than the treatment. And there were no major adverse events. And this is the phase 1 trial outcome. So, again, no major adverse events. The minor events weren't, really something to worry about. And our target plasma concentration, at 300 nanograms per mil, from that we got guidance on from the Delitecchi study, we were able to hit that at the 500 milligram dose level. So you can see that here we've got, this is the highest dose level, but even here, we were able to hit that exposure and this is now in humans. So well-controlled study. And we have all kinds of information from this trial. We can reasonably dose. We're looking at spinning this into children and seeing how we can do that. So it was a useful study that was actually finished March of this year. The food effects studies here, we gave the healthy volunteers a FDA standard high-fat breakfast, 900 calories, over 50% of calories from fat, butter. The eggs were cooked in butter, cream cheese on your bagels, that kind of stuff. And you can see that actually with the high-fat diet, we got a blunting of the exposure, which was actually the opposite of what I was expecting. I was expecting that lipophilic environment from the high-fat diet would help exposure, help get it past the liver, but in fact, the opposite is happening. And it looks like it's probably disrupting our micellar structure, which, when we did the formulation, we paid quite a lot of attention to the micellar size and those sorts of things. So this will probably result in a labeling on the drug to take on an empty stomach with a glass of water, something like that. We have, and so that's where we are. We have our phase 2 trial designed. We have, right now, we're looking at a double-blinded crossover design. It's a rare disease, so getting a hold of patients will always be a challenge. We're looking at something like 10 to 20 MPS I patients per group, given Jutraw and then crossed over to placebo after a washout period. Patients will be allowed to continue ERT or else we would never fill the trial. And the endpoints are, like I said, range of motion, quality of life, and pain survey. We are going to measure the 6-minute walk and forced vital capacity, which were the basis for the approval for the enzyme replacement therapy, because the FDA wants us to measure that. So, okay, fair enough. But we really hope to get an outcome based on this. Actually, recently, we've started to look at quality of life assessments from the patient, but now also incorporating a caregiver quality of life survey, where you have a parent of a child and they see their child is much more active, for example, and they will be able to report that possibly better than a child. So this is sort of a new emerging trend in the lysosomal storage disease area that we're going to try to take advantage of for our phase 2 trial. We're going to do phase 2 trials also for the Alzheimer's indication and for the Friedrichs ataxia indication as well. Those are also being planned. And more in the academic realm, I want to sort of leave you with the idea that we can do this. It's not so hard to test a bunch of compounds, repurpose compounds for their ability to increase a deficient enzyme, those sorts of things. And so even with the Lynx L1000 data, we are able to now, this is for the MPS II enzyme, we're able to pick out compounds that will upregulate that enzyme, even without having to do screening. So these are the kinds of ideas that we can now further implement in order to find additional compounds down the line. And that's all I have to say. Leave a little bit of time for questions. I definitely want to thank my colleagues, collaborators, and all of the funding we got for it, and especially the Ryan Foundation. They were very supportive. I know there was one question, but I'm happy to take any additional questions or discussion. Thanks for your attention.
So James asked a question about does Resveratrol decrease GAG accumulation in lysosomes? So we have not measured that, and I guess I'll say the number of things we haven't measured definitely outranks the number of things we have measured. We try to stay hyper-focused on whether we're getting the primary outcomes that we want. So no, we haven't measured that. We haven't even measured any glycosaminoglycan accumulation. The Patient Foundation actually was the one who advised us against measuring GAGs because of the high variability of the glycosaminoglycans in the patient population,
could lead us to a negative answer. So for example, we might not get a change in GAGs with resveratrol. So they basically told us that, you know, it wasn't a worthwhile endeavor to look at that. So we really haven't done anything beyond really this hyper-focused clinical approach that you've seen.
I was wondering about the quality of life surveys, either from the patients or the patient families. That just seems like a really complicated thing. And I would imagine you would need a large sample in order to be appropriately powered. Do you have anything to say about that?
Yes, well, so you're right. For the most part, I would say some of the domains that are on that quality of life survey are so impacted by the disease. And I'm talking about things like, you know, these patients can't open a fridge because they're arthritic. And it's
Nancy, could you please mute your phone?
I'm so sorry.
Thank you.
So some of these, you know, domains are very impacted by that. So in some ways you could say, well, there's a great window for improvement. And keep in mind, it's a phase 2 trial. So what we're really looking for are what are the domains that are really going to be affected by resveratrol, and what are the domains that we can kind of ignore in a phase 3 efficacy trial. So that's kind of where we're at. We don't exactly know what's going to happen, and so we're doing a little bit of guessing and that sort of thing.
I have another question, and I kind of know the answer to this question, but I don't think everybody on this call knows the answer. Can you tell us what a Lynx 1000 assay or study is?
I'm actually probably the least qualified. I had a postdoc, Zach, and he went and figured it out. Lynx 1000, as I understand it, anybody who knows better, please jump in and save me. As I understand it, is essentially it's a bioinformatic effort to take data that has already been generated based on cells that have been treated with different compounds. And, you know, you can imagine how many different labs across the world have ever tested compounds on cells. And this effort is bioinformatically takes all of that cellular outcome for 1,000 different compounds, combines it, and sort of creates an average of what the effect is on the compounds. And apparently it's a very powerful tool, and I don't know how good it is, but everything that I've heard, it's quite good. And Stefan Schur here at University of Miami sort of runs that effort for the University of Miami site, but it's a nationwide effort to do this. I hope I didn't butcher that.
So I think that's close. So the 1,000 doesn't refer to compounds. It refers to genes that are markers for pathways that are supposed to represent the genome. And so they have a very inexpensive way of measuring the expression of those 1,000 genes. And then they use those as signatures. And then many, many, many more than 1,000 compounds have been run on many different types of cells. And you get these signatures, and so you can use that as a background to compare your treatments on your cells and see if maybe you can identify pathways that have been identified with other well-known drugs. Yeah, it's a powerful technology. Does anybody else have questions?
Shaun, I had a question, Dalton. Great talk. Thank you very much. I really enjoyed it. In terms of the dosing regimen that you're currently in the phase 1 trial, does the dosing go for 12 weeks? Can you just tell me, maybe I missed that?
So in the phase 1 trial, we merely measured pharmacokinetic exposure, and that will set us up for the phase 2 trial to really do the prolonged exposure. We haven't done any of that yet. The Murdoch Children's Research Institute and the Turner trial, those were both phase 2 trials. So there is some knowledge about chronic dosing of resveratrol, but that is what also part of what our phase 2 trial is going to be is a longer term dosing of resveratrol to try to achieve an effect. So, you know, phase 2 trial, for those of you who don't know, isn't an efficacy trial. We're not expecting to get a clinical benefit in a phase 2 trial. It's really a trial to set up for the phase 3 trial, which is the clinical efficacy trial. So the phase 2 trial sort of determines what the dosing should be, you know, whether you have enough patients, whether you have enough power, and whether you're getting any additional toxicity, especially in the patient population, that would prohibit going to a phase 3 trial.
Yeah, but I think more people are emphasizing phase 2 trials, larger phase 2 trials, instead of going to the phase 3. Maybe it's different in a deficient enzyme type of a problem. But so the hypothesis is that the treatment can elevate, some of the activity of the enzyme, but that over time you have to continue to give the drug. Is there any evidence that at some point there could be some metabolic plasticity where the enzyme changes its phenotype, but that's not the case, right?
Yeah, I mean, all of those are outstanding questions. We feel that, yes, a chronic treatment, daily administration of the drug will be required for all the patient populations I've talked to you about. Resveratrol is an interesting molecule. Not only does it, so there's a lot more complication to this, and I won't go into it, but not only does it increase iduronidase levels, it's also anti-inflammatory, has anti-inflammatory properties. And it's probably the anti-inflammatory properties that are what the Alzheimer's trial is really picking up. And so you can imagine that in a population like MPS I, where they have inflammation, that could also provide a long-lasting benefit. And in fact, our clinical trials outcomes with pain and range of motion will actually also pick up any anti-inflammatory activities. So there's more. And then, reactive oxygen species are it's a scavenger for ROSs and things like that. So there's many, many more activities than, this very simple thing that I'm showing you. And yeah, I would imagine chronic dosing is definitely something that will be required. And then I guess just to fill that out, with the enzyme replacement therapy, that's administered once a week. So we feel like if we can hit the enzyme 7 times a week with once daily dosing, that will be better than once a week that you see in the enzyme replacement therapy, because GAGs are slow to build up. They don't build up quite as fast, and it's more of a chronic disease. So we feel like whatever we do more chronically could be better than the enzyme replacement therapy.
Any other questions?
Thank you guys so much for having me. I really look forward to working more with you. If you guys have any interest in any of this, I'm happy to discuss it more.